A keratinocyte is the predominant cell type in the epidermis, the outermost layer of the skin, constituting 90% of the cells found there. Those keratinocytes found in the basal layer, stratum basale, of the skin are sometimes referred to as basal cells or basal keratinocytes. Function. The primary function of keratinocytes is the formation of a barrier against environmental damage by pathogenic bacteria, fungi, parasites, viruses, heat, UV radiation, and water loss. Once pathogens start to invade the upper layers of the epidermis, keratinocytes can react by producing pro-inflammatory mediators, particularly chemokines such as CXCL10 and CCL2, which attract leukocytes to the site of pathogen invasion. Structure. A number of structural proteins, filaggrin, keratin, enzymes, proteases, lipids, and antimicrobial peptides, defensins, contribute to maintain the important barrier function of the skin. Keratinization is part of the physical barrier formation, cornification, in which the keratinocytes produce more and more keratin and undergo terminal differentiation. The fully cornified keratinocytes that form the outermost layer are constantly shed off and replaced by new cells. Cell differentiation. Epidermal stem cells reside in the lower part of the epidermis, stratum basale, and are attached to the basement membrane through hemidesmosomes. Epidermal stem cells divide in a random manner, yielding either more stem cells or transit amplifying cells. Some of the transit amplifying cells continue to proliferate, then commit to differentiate and migrate towards the surface of the epidermis. Those stem cells and their differentiated progeny are organized into columns named epidermal proliferation units. During this differentiation process, keratinocytes permanently withdraw from the cell cycle, initiate expression of epidermal differentiation markers, and move suprabasally as they become part of the stratum spinoceum, stratum granulosum, and eventually become corneocytes in the stratum corneum. Corneocytes are keratinocytes that have completed their differentiation program and have lost their nucleus and cytoplasmic organelles. Corneocytes will eventually be shed off through desquamation as new ones come in. At each stage of differentiation, keratinocytes express specific keratins, such as keratin 1, keratin 5, keratin 10, and keratin 14, but also other markers such as involucrin, loricrin, transglutaminase, filaggrin, and caspase 14. In humans, it is estimated that keratinocytes turn over from stem cells to desquamation every 40-56 days. Whereas in mice the estimated turnover time is 8-10 days. Factors promoting keratinocyte differentiation are: a calcium gradient, with the lowest concentration in the stratum basale and increasing concentrations until the outer stratum granulosum, where it reaches its maximum. Calcium concentration in the stratum corneum is very high, in part because those relatively dry cells are not able to dissolve the ions. Those elevations of extracellular calcium concentrations induces an increase in intracellular free calcium concentrations in keratinocytes. Part of that intracellular calcium increase comes from calcium released from intracellular stores, and another part comes from transmembrane calcium influx, through both calcium-sensitive chloride channels and voltage-independent cation channels permeable to calcium. Moreover, it has been suggested that an extracellular calcium sensing receptor, CASR, also contributes to the rise in intracellular calcium concentration. Vitamin D3, cholecalciferol, regulates keratinocyte proliferation and differentiation mostly by modulating calcium concentrations and regulating the expression of genes involved in keratinocyte differentiation. Keratinocytes are the only cells in the body with the entire vitamin D metabolic pathway, from vitamin D production to catabolism and vitamin D receptor expression. Cathepsin E. Tail homeodomain transcription factors. Hydrocortisone. Since keratinocyte differentiation inhibits keratinocyte proliferation, factors that promote keratinocyte proliferation should be considered as preventing differentiation. These factors include the transcription factor p63, which prevents epidermal stem cells from differentiating into keratinocytes. Vitamin A and its analogues. Epidermal growth factor. Transforming growth factor alpha. Cholera toxin. Interaction with other cells. Within the epidermis, keratinocytes are associated with other cell types such as melanocytes and Langerhans cells. Keratinocytes form tight junctions with the nerves of the skin and hold the Langerhans cells and intradermal lymphocytes in position within the epidermis. Keratinocytes also modulate the immune system. Apart from the above-mentioned antimicrobial peptides and chemokines, they are also potent producers of anti-inflammatory mediators such as IL-10 and TGF-beta. When activated, they can stimulate cutaneous inflammation and Langerhans cell activation via TNF-alpha and IL-1 beta secretion. Keratinocytes contribute to protecting the body from ultraviolet radiation, UVR, by taking up melanosomes, vesicles containing the endogenous photoprotectant melanin, from epidermal melanocytes. Each melanocyte in the epidermis has several dendrites that stretch out to connect it with many keratinocytes. The melanin is then stored within keratinocytes and melanocytes in the perinuclear area as supranuclear caps, where it protects the DNA from UVR-induced damage. Role in wound healing. Wounds to the skin will be repaired in part by the migration of keratinocytes to fill in the gap created by the wound. The first set of keratinocytes to participate in that repair come from the bulge region of the hair follicle and will only survive transiently. Within the healed epidermis, they will be replaced by keratinocytes originating from the epidermis. At the opposite, epidermal keratinocytes can contribute to de novo hair follicle formation during the healing of large wounds. Keratinocytes migrate with a rolling motion during the process of wound healing. Functional keratinocytes are needed for tympanic perforation healing. Sunburn cells. A sunburn cell is a keratinocyte with a pycnotic nucleus and eosinophilic cytoplasm that appears after exposure to UVC or UVB radiation or UVA in the presence of psoralens. It shows premature and abnormal keratinization, and has been described as an example of apoptosis.
Please subscribe and thanks for watching.